Welcome to Creation in the 21st Century. I'm your host, Carl Baugh, founder and director of the Creation Evidence Museum in Glen Rose, Texas. The title to today's program is Designer Genes. Now, that creates tremendous illusions and considerations of the mind. For instance, we are standing in an incredibly gorgeous replication of the Palace of Shushan, Ahasuerus Palace, and it certainly was designed. Even the replication was designed so that the roof fits, the columns fit, and if we want to move something over, everything has to be able to support the entire matrix and construct of the total design feature. But this marvelous and spectacular replication of Shushan Palace is nothing compared to the design within your human body. And within every cell of your body, you have the DNA in the nucleus, the deoxyribonucleic acid, and that little tiny information library does incredibly wonderful things. Designer genes are in vogue, and I've brought today to the studio a dear friend of mine, Professor John Hefner, well qualified to discuss this issue. Professor, it's always a privilege to have you on the program.
It's always a pleasure to be here.
You've been here many, many times.
Quite a few.
And you have almost 40 years, almost 4 decades as a career specialist in mathematics.
Yes.
You teach pre-calculus, trigonometry, and all the nice things that give unique direction and confluence, not only unique direction, but confluence to constructs. And that's what we're going to talk about today in genes. When I first heard you talk about designer genes, I thought you were talking about the fashion genes that our teens are involved with.
Well, I did a play on words, you know. Designer genes is on a lot of people's mind, but I'm going to spell it with a G.
Rather than a J. All right. Isn't this, now we talk about designer genes that are quite simplistic but necessary, designer genes for the fashion market, for clothing. Neat idea. But incredibly simplistic compared to the designer genes in our bodies and in the nucleus of the cell of all living systems. 4 basic little elemental particles that can be separated so that you can form a second replication. In fact, you and I started as one cell.
Yeah.
But in that cell was all the information necessary to replicate and program eyes, hair, ears, nose, feet, toenails, teeth, everything. What an incredibly beautiful concept.
It is.
I wonder who thought of that.
Oh, well, I think I have an idea.
Well, yes. Oh, yes. There had to be a designer for all of that.
Yes.
And aren't you glad it didn't have to experiment so we would have an arm running out our back for a while?
Oh, yeah.
And then he would decide, that won't work, so give him 3 feet instead. Well, that won't work. Instead, the design, even of the human locomotion you and I will use on the set today, that design to swing in perfect balance on a pelvic structure is all encoded within those designer genes.
Oh, it's an unbelievable litany of features that have to be, have instructions for that to occur.
Yes, and all has to work simultaneously.
And it's interesting, no one would suggest that designer blue jeans just happen by the wind blowing across a cotton field and the fibers hanging up on a barbed wire fence and weaving into fabric that is fashionable. No one would believe that. And yet some are willing to maintain that something far more complex and functional,
infinitely more complex,
just happened by chance, time and chance, and, you know, beneficial mutations, which are really birth defects.
Sure. A foolish, very foolish philosophic approach to how we got here and who we are, and the brain itself, along with all of these other features, all involved in the designer genes. All right, take us on the road. I want to hear what we have to learn today.
Well, first I thought it would be appropriate to just cut some ads of blue jeans out and put the little play on words, designer genes.
Sure.
But just a quick little brief history. In 2001, the human genome was found to be far more complex than previously thought. Scientists, in fact, are amazed that the human genome was reported to only have about 35,000 genes. Now, previous estimates were 4 times as much as that, 140,000 genes.
And our first thinking was, well,
Yes.
wait a minute, it's not quite as complex as we thought.
Yeah, so at first thought, we think, well, you know, we're not as complex as we thought we were. We only have 35,000 genes. Well, but actually this vastly lower number means that the human genome is more complex.
Yes.
Because these genes can be read forward and backward and even three-dimensionally.
And the so-called junk DNA
Oh, yeah.
responds to unique environmental context. This is an incredibly complex and incredibly beautiful design for living systems.
That's true. And in fact, in 2004, the International Human Genome Sequencing Consortium, it's a long mouthful of words, but they reduced it down to about 25,000 genes. And
That means they're more complicated.
Yes, and they said, and their quote is, "It blows our socks off," as reported in Nature magazine back in 2004. "We have a long way to go to understand how all the parts revealed by the genome sequence together to make life." And what we're finding is that these things are obviously multitasking.
So they're much more complicated than we had originally thought.
That's right, and they are read, like it says, forward and backward and three-dimensionally. I think of it as like a notebook of transparencies with words written on the transparencies, but as you read down vertically through the stack of clear transparency sheets, what if the letters stacked up made words and meanings being read both ways and backwards and forwards and up and down and all of that? And that's probably a pretty close analogy of what we actually find
Beautiful analogy, yes.
in the human genome. Well, we've all got our own set of designer genes. That's the point you made so well. An example of multitasking genes, which we're discussing, university...
Now let's pause for a moment.
Okay.
Let's define genes, Professor. What are we talking about?
Well, I have a little thing coming up that's going to kind of make an analogy with letters,
the audience, yes,
words,
let
and
us know.
sentences.
Sure, what the genes actually are.
And I've got that coming up, but just suffice it to say it's a complex little piece of information that carries out a function. And for example, the CX26, which is a gene associated with deafness or hearing, get this, also plays a role in helping wounds to heal.
Now that's incredible.
Seemingly unrelated features, but again, a multitasking gene.
And depending on how it's read forward, backward, or three-dimensionally.
Evidently.
Yes.
And this is their report. I just know what I read in the paper, you know.
Yeah.
It's impossible to know the precise value of a gene. There's value in assuming that inconvenient but survivable genes might be there for a reason.
Oh,
as reported by Discover magazine. Now, in complete honesty, they mean there for an evolutionary reason, but we think there's a reason, all right, but it's a design feature.
It's a design feature.
Rather than some accidental feature. Now, a question you just raised is about junk DNA. And here's the answer about so-called junk DNA. It's possible that non-coding DNA contains signals that decide when other genes are switched on or off. They're not necessarily always on, as reported by Science News back in December of 2005.
And this audience needs to know that between Science, and Science News repeats it from Science, between Science magazine and Nature magazine, we have the world's 2 most prestigious scientific journals. So this is not simply an afterthought of some incidental scholar. These are major statements by major microbiologists and biochemists.
That's right. And the non-coding region of the genome, once known dismissively as junk DNA, is crucial, as reported in Time magazine in 2006.
That's a major statement.
It's major because they wanted to, it seems like they did, to dismiss all of that to junk DNA that was reflective of our evolutionary history.
Yes.
And relegated to some, you know, forgotten warehouse somewhere in the body,
yes.
in essence.
The idea, as you said, was that in our evolutionary history, we accumulated a lot of genes, a lot of junk that we don't need, a lot of glob that we just really don't need.
And what we're finding is that it's functional.
It's absolutely crucial for the functioning of the entire system.
It's an amazing system. In 2002, the German evolutionist Svante Pääbo was reporting how amazingly complex these genes are. He found only tiny differences in the chimp versus human fox P2 gene, which is located in the so-called junk DNA.
Okay.
And they thought, you know, it didn't do anything, the evolutionists did, but it actually controls the speech center of the brain.
Which the chimpanzee does not have.
That's right. But, you know, there's only 2 amino acids different between chimps and humans.
In that particular gene.
Which really isn't enough to differentiate why we can talk so fluently, and they essentially are mute, just a few, you know, squeaks and chirps and grunts and growls. And Pablo says it's the gene expression that makes the difference. And I'm not sure I fully comprehend what is involved in that term expression. It's not merely the genes. He said 10% of the human versus chimp genes studied are expressed at significantly different levels in the 2 species. So evidently that is a key. There's a lot more difference between humans and chimps
Absolutely.
than they have been willing to admit to, it seems.
Certainly.
So for a new trait to evolve, you can't just have one gene mutate.
Right.
The regulatory genes that control it would have to mutate as well.
And simultaneously.
Exactly. And this gets way too complex to even imagine how it could have possibly happened. And
Dr. Charles Jackson, our mutual friend, who's been on this program, by the way.
And here's a little diagram of the double helix molecule of the DNA. And it's really a very clever system, you know, with the information on one strand and then the other strand checking it and cross-checking it and so forth. Somewhat of a redundant system.
What an incredibly beautiful and splendid design. No leftover parts. We have 4 elemental components, guanine, thymine, cytosine, adenine. Now, those may be strange words to many in the audience, but just 4 basic elemental components which by themselves say nothing. But in combination, they express proteins that make us function. And this is the library index of information. And yet, as we talked at the top of the program, we began as a single cell with a single grouping of this in the nucleus. Yet, it has written into it, encoded into it, the beautiful ability to divide and replicate, the other side being replaced for both of them. Then you have 4 sides or 2 cells.
Well, it minimizes the effect of destructive mutations. It doesn't totally eliminate it, as we'll soon see, but it's a good system nonetheless, as long as the data is not corrupted in the first place.
So these genes are actually the encoding of information that makes that physical body function at the basic level, at the library level.
Well, these are the nucleotide bases, yes. Now, the Institute for Creation Research, very fine research organization, as you know, out in California. A recent article by Professor Daniel Criswell entitled "The Code of Life, Little Words, Big Message," very amply titled. The genes are polyfunctional. He's reiterating that concept, able to produce more than one protein depending on which direction the gene sentence is read or where the gene starts and stops. They help regulate the amount of protein produced by the cell and possibly assist in folding it into the correct shape, functioning shape. DNA is the blueprint, RNA is the contractor, protein is the house.
Oh, yes.
So that's a good analogy.
Good.
And these letters, these 4 bases here, usually labeled by their first letter, you know, A, C, G, and T.
Yes.
For the 4 nitrogenous bases, it says they're arranged in a chain with 2 strands forming a double-stranded molecule. One strand has the coding information, and the complementary strand is used as a template to correct damage or mutations in the coding strand,
Isn't that beautiful?
Just mentioning. So as I began to be interested in this and start to read it, I ran across a very wonderful book by one of the leading experts and scholars in the world, Dr. John Sanford, previously at Cornell University. True scholar, Genetic Entropy and the Mystery of the Genome. This is Dr. Sanford here in his book. He's the inventor of the gene gun.
Of the gene gun.
And quite famous, has founded 2 companies, you know, and so forth. And this is a picture here of him. And of course his bio is pretty legendary. But down here he says, respected Cornell University geneticist John Sanford rejects Darwinism in his recent book I mentioned. And a quote, "The issue of who we are, where we came from, and where we're going seemed to me to be of enormous importance." And I was really taken by this true scholar's sense of perspective of where he is in the timeline of life and things that really matter. He says, "In retrospect, I realize I've wasted so much of my life arguing about things that don't really matter, it's my sincere hope that this book can actually address something that really does matter. So I was even more interested to read it.
And it's a brilliantly written book.
And the whole premise, he says, modern Darwinism is built on what I'll be calling the primary axiom. And here it is. The idea that man is merely the product of random, I might say accidental, mutations plus natural selection.
That's the concept of evolution,
and I
heart
couldn't say
and
it any
soul.
better. A constantly mouthed mantra repeated endlessly on every college campus.
Yes, just repeated. No substantiating evidence, but just repeated. And it's the mantra. It's the dogma.
And the primary axiom is actually an extremely vulnerable theory. In fact, it's essentially indefensible. Its academic invincibility derives mostly from bluster, smoke, and mirrors. Many college students have certainly seen that. If you dare question evolution in the classroom, you're often met with scorn and wrath and intolerance and intimidation.
Normally, yes.
And threat of grade damage, no telling what all. And this axiom is held up by an almost mystical faith, he says. This man ought to know. He's been on the college campus for decades.
And one of the great inventors.
Exactly.
And a former evolutionist and professor himself.
Right. And it says, which true believers have in this, this mystical faith that they have in the omnipotence of natural selection.
Yes.
And Professor Sanford said he now has a very strong conviction that the primary axiom is most definitely wrong. I say it out loud, the emperor has no clothes.
No clothes. It won't fly, just won't fly.
So by the genome, you had asked earlier, the genome is really a somewhat overarching term that includes the chromosomes, the genes, and the nucleotides. You know, those four bases that we mentioned earlier.
All of this
and the nucleotides,
basic structure.
In fact, this is kind of a graphical representation of those bases in sequence, not unlike letters that form words, that form sentences, that form paragraphs, that form chapters, that form books, that form libraries. And those same analogies are right here. The nucleotide bases being the letters, the motifs or clusters are the words, the genes are the chapters, chromosomes are the books, and the genome is the library.
Total genome is the library.
Yeah.
Now, in these last moments, you're making a profound statement that these genes have been designed. And they were, and of course we'll get to the designer. So can you give us, in a nutshell, the statement that you're making?
Well, in addition, he says, to multiple overlapping linear language-like forms of genetic information, the genome is full of countless loops and branches going different places. The genes that regulate, it has genes that regulate genes that regulate genes
that regulate genes.
And again, even the DNA we found, it folds into various shapes, similar to amino acids, proteins that fold to fit like a key going only into one lock.
Yes.
And our bodies, we must remember, contain over 100 trillion cells. And each one has a complete
set of instructions.
Isn't that amazing?
Amazing.
And the mystery is, he says, where did all this information come from and how can it possibly be maintained? It's like a manual of who we are.
Yes.
And the standard evolutionary answer is, well, mutation is where it came from, and natural selection selected those mutations that were beneficial, that conferred some advantage to the organism. And they've created all this biological library of information. Life is life because random mutations at the molecular level are filtered through a reproductive sieve at the level of the whole organism.
Now that's the standard answer. But, as Dr. Sanford reiterates in his book and in person, that is a mantra that's repeated and repeated and repeated without substantiation. The emperor has no clothes. It just won't fly.
That's right. So I made a slide here. We've all got holes in our genes. I put holes in the knees.
Okay. Now give us the answer. Where did it all come from, all of this wonderful information?
Well, he says essentially, if you could show the primary axiom, the foundation is wrong, then our whole current understanding of life would also be shown to be wrong. The current evolutionary understanding.
Yes.
Well, newsflash, random mutations consistently destroy information.
That's
what
right.
the audience needs to remember is that all mutations always result in a loss of information.
That's right.
And evolution needs a net gain. If you're going to go from pond scum to Einstein, you've got to have some additional information for arms, legs, eyes, ears, brain, things like that. And they do believe that, essentially, it's the story of evolution, and you've got to have that increase in information.
They believe it, but it's never been observed and never demonstrated in the laboratory or the field experiments.
Well, and how can losing information all along the way give you a net gain?
You're a mathematician.
Yeah, so it's just not going to fly mathematically or any other discipline of science.
Right.
And the strong predominance of deleterious mutations absolutely guarantees the loss of information from the genome.
Yes.
And furthermore, he says there's so many different types of mutations. There's what's called point mutations, like one letter out of place, like a typo. There's deletions where you leave one out. There's insertions where something extra is in there. There's duplications that shouldn't be there. There's translocations. There's inversions, and then there's mitochondrial mutations in another part of the gene of the cell from the nucleus. So,
and this is all harmful or deleterious.
that's right. And we talked about junk DNA, and now we know that most of it is functional. It appears to be written in both directions, so he says it's doubly functional, as one of our other scholars that I quoted earlier
Now
says.
three-dimensionally, triply
And so
functional.
if it's doubly functional, then a mistake is going to be even more harmful, because it harms it from what was going to happen in either direction.
Yes, certainly not beneficial.
And the bad news is we are on a downward slide that cannot be stopped. Now, isn't that a bold statement? Now, someone may be tempted to say, but wait, won't natural selection take care of this problem? The answer is no, he says. There's an enormous chasm that exists between genotypic change, which is molecular mutation, and phenotypic selection, which is the whole organism.
Whole organism in its reproduction.
Yeah. So one little gene, you don't select the individual genes and then build the organism you want. You take the package.
Yes.
And when Mother Nature selects for or against an individual, she has to accept or reject the whole organism. And as I said, it's a package deal there.
Package deal.
And he made the analogy like you're selecting a TV, and you don't go and select the individual pixels and then build your set. You basically you purchase the overall best picture.
That's right.
And so that's essentially what he's saying in these sentences here as we close, you know, that you have to take the whole package. Now, other scholars agree with Dr. Sanford, and they know that just one additional mutation per generation is going to take us downhill.
Ultimately lead to extinction.
Yes.
Not the production of a
Dr.
new.
John Morris, very fine scholar at ICR, says the current count of human gene disorders, and he names a few like Alzheimer's, sickle cell anemia, muscular dystrophy, and so on, is around 13,000. Now, not everyone has all 13,000, but the human genome somewhere,
Yes.
we have those.
And we have many of them personally,
In the human gene
whether
pool.
expressed or not. Yes.
That's right. And Dr. Joseph Mastropaolo, who's also been here and
That's
is an
correct.
expert,
Fine scholar.
he gave the count of known human genetic mutations, and we're, you know, somewhere beyond 10,000 now.
Let's take just a moment, even though time is fleeting, let's take a moment. In 1960, we knew of 1,000 genetic disorders. 1970, 2,000. 1980, 3,500. 1990, 5,000. 2000, 10,000. By 2010, since we already have 13,000 known genetic disorders, it's projected we'll have 30,000. 2020, 60,000, 2030, 100,000. By 2085, at the rate at which we're going, we're not evolving. This audience needs to know we're not progressing. We are headed toward extinction if the Creator doesn't come back and correct our problem. And as you stated, and as Dr. Sanford stated, the scientific conclusion is mutations destroy life. They do not create life.
Yes, and that destroys the primary axiom upon which evolution is built.
Yes.
And so I was gratified to see that in his concluding remarks, he said, even as we cannot create life, nor can we defeat death. In essence, it's been said, everyone is on death row. You
Yes.
know, we are approaching
Genetically.
an appointment with our Maker.
But he provided an answer.
He says, yet I assert that there is one, with a capital O,
Yes.
who did create life and who designed the genome. He's called the author of life, Acts 3:15, and I believe that the author of life has the power to defeat death and degeneration. I believe that is the good news.
Let's emphasize that good news for the audience. Dr. Sanford has been quoted heavily by Professor Hefner today, and with absolute justification. Dr. Sanford defected from the camp of the evolutionists, even thought atheistic and agnostic thoughts in his life. Former professor, the inventor, in fact, is still a professor, inventor of the gene gun, one of the world's leading microbiologists, and he was schooled in and taught and accepted evolutionary theory. But when he got to looking at the nitty-gritty, and that was his specialty, he found that at the basic level, Evolution does not produce higher orders. It loses the ground on which it stands. Now, he concluded with a statement, and Professor Hefner and I agree with this. There is one who created life and has the ability to correct life. Not only will he one day refashion us so that we will be made in his image completely, physically and in every dimension, but he also gave us an incredible answer to the deeper problem, and that's the problem within our soul we call the sin problem. Jesus Christ visited planet Earth. Our Creator was made flesh, walked among us, went to Calvary, died for us, was placed in a cold, dark tomb, and arose from the dead. At this moment, that living Creator, Savior, is knocking at your heart's door. Would you just pray this simple prayer as you open your heart to Him? Just pray this prayer with me. Dear God, I'm a sinner. I have a problem. I'm headed toward annihilation, extinction, loss of life, but with an eternal destiny without you, hopeless. I need you. Lord Jesus, right now, I ask you to come into my heart. Cover me with your blood. Forgive my sins and give me life that you provided. I receive you as my Savior right now. Come in, Lord Jesus. If you prayed that prayer, recognizing yourself as a sinner and Jesus as the answer, then He covered you with His blood. Heaven is your home, and ultimate total repair is your destiny.
Creation in the 21st Century has been sponsored by Trinity Broadcasting Network, and only with your love gift of support can this program stay on the air. So write to Creation in the 21st Century, P.O. Box A, Santa Ana, California, 92711.
Creation in the 21st Century is a unique program on TBN combining biblical knowledge with scientific verification. Much of the information that I use on the program is available. Contact us. Just write Creation Evidence Museum, P.O. Box 309, Glen Rose, Texas 76043, or call us at 254-897-3200. We look forward to hearing from you today.